Good afternoon. My name is Andrew Crockett. I'm the CEO and one of the co-founders of KalVista. I'm pleased to be with you here today to talk to you about our company and give you an overview of what we're up to. Just a reminder that the presentation does contain forward-looking statements. I'll refer you to information included on this slide. So in terms of highlights, as a company, we've been focused on discovery and development of small molecule protease inhibitors since our inception. We're currently focused on small molecule plasma kallikrein inhibitors, oral inhibitors for hereditary angioedema, as well as intravitreal and oral inhibitors for diabetic macular edema. Our lead molecule in HAE is KVD900, which is currently in a phase 2 clinical trial for acute therapy of attacks of HAE with data expected in late 2019. For our programs in DME, we have a collaboration with Merck. Our lead program, KVD001, has a phase 2 trial that will read out in the second half of the year. We also recently started a first-in-human trial with KVD824, and we will give an update on this program in middle of the year. I'll speak more about that in a minute. At KalVista, we do all of our own chemistry, all of our own drug discovery and development, and so when we think about IP, that's critical to us, and it's mostly composition of matter with other ring fence around those compositions and those compounds. The company's well funded. We have reported just over $111 million as of January 31st, and we're funded into 2021. Here's a snapshot of our product portfolio. I referred to both KVD900 and KVD001 before. These are our programs that are both in phase 2 clinical trials. I'll touch on both of these again later in the presentation. KVD824 is the program we just moved into a first-in-human trial. This will be positioned either for prophylaxis of HAE or as an oral treatment for DME, depending upon the data we see from the phase 1 trial. We'll give an update on 824 in middle of this year. We will continue to bring additional compounds forward, plasma kallikrein inhibitors. Again, I mentioned 2 indications, additional indications we're interested in, both in prophylaxis of HAE as well as in oral compounds for DME. And then we're working on additional molecules for targets that are undisclosed. So a brief background on hereditary angioedema. This is a genetic condition that leads to painful and dramatic events of swelling in various parts of the body. It is an orphan disease. The incidence is 1 in 10,000 to 1 in 50,000 persons. And it's primarily caused by a defect in C1 inhibitor activity, which leads to uncontrolled plasma kallikrein activity and excessive release of bradykinin, which is a mediator of edema in HAE. There are multiple approved products targeting plasma kallikrein now, many well-controlled clinical trials, and actually all the approved products in this space work along this same pathway, either through C1 inhibitor, plasma kallikrein, bradykinin, what have you. So they're all along this pathway. It's a very well-validated target. But all those currently approved therapies are injectable, and so we believe that leaves a high unmet need for oral therapies. This is a very mature commercial opportunity for a rare disease with over $2 billion forecasted by 2020. And as I alluded to earlier, we intend to bring candidates forward both into the acute and prophylactic segments. Just a quick review of kind of where we stand with the market. The on-demand segment is the largest part of the market. Firazyr is the leading compound here. One certainly important evolution in the market that's just kind of, we're in the middle of it, frankly, is the launch of Takhzyro on the prophylaxis side and the growth of Haegarda as well on prophylaxis. We think that oral therapy, as I mentioned earlier, represents a significant opportunity and advancement for patients in the way they treat this disease. It's certainly what patients want and demand, and is a really important factor as they think about the future of this market. Even patients that are well managed on prophylaxis require acute therapies at all times to be with them. So again, if you think about the acute opportunity, it's not only for those patients who desire that as their first line of therapy, but also desire it as an add-on or for breakthrough therapy in prophylaxis. I'll now review with you some data that we released last summer with our lead compound in HAE and acute treatment of HAE. This is KVD900. This trial was in healthy volunteers. It was a double-blind randomized investigation of KVD900 in 84 healthy male volunteers. We had a single ascending dose phase of the study. We then also crossed over from a capsule to a tablet. So we'll look at some of that data as well, and then also did a food effect part of the study. And we'll review all this data, safety, tolerability, pharmacokinetics, and pharmacodynamics as we move through the presentation. Here's the safety data from the study. These are all the adverse events that we saw in the study. I think some kind of important highlights: there were no SAEs. Of the 45 adverse events we saw, 44 of them were mild. The one moderate adverse events we had was a headache in the lowest dose group, in the 10 milligram dose group. Importantly, we had no GI adverse events considered related to KVD900. That's a really important point, especially as we consider some of the other products moving forward in this space. That absence of GI adverse events, something that we were focused on. There was one incident of vomiting that occurred 3 days post-dose, so we certainly wouldn't have that be related to the drug. And as you can see in the single ascending dose phase, you see two columns that are blank at the 3 and 600 milligrams, and that's intentional, so that there were no adverse events at those higher doses, which is certainly great to see as we think about moving forward. Here's the PK profile of 900. These are the capsules. And what you can see is dose-dependent exposure, both in Cmax and AUC, and very rapid and high exposure of drug, a mean Tmax of around an hour, and in the capsules, a mean Cmax of 3,500 nanograms per milliliter to 600 milligram dose. So really a profile that we intended for acute, that is rapid and high exposure of drug. I mentioned earlier we did a crossover from a capsule to our tablet formulation. So here we can see in the green line, this is the capsule data, the 100 milligrams, similar to what you saw on the previous slide. And what you can see is in the blue line is our tablet is performing even better, so getting in quicker and to higher levels than our capsule formulation. This will be our commercial formulation moving forward. We also looked at food effect on absorption and inhibition of the drug. And what we can see here in the bottom panel is that effectively the fed status had no impact on the pharmacodynamics, with KVD900 tablets delivering 95% inhibition within 30 minutes in both a fed and fasted state. Here's some additional data looking at two doses of with our tablet, 100 milligrams and 600 milligrams. This is both pharmacokinetics and pharmacodynamics. What we see is very strong inhibition of plasma kallikrein, almost complete inhibition of plasma kallikrein following administration of both of these doses. This is the assay here is a plasma kallikrein activity, which is monitored using a fluorescent substrate. The plasma is activated by the addition of dextran sulfate. So this is kind of a standard assay. We do this in whole plasma, which we think is important because it's relevant to kind of the physiological state. And what you can see here is, again, is both doses reach complete inhibition of plasma kallikrein. The difference is the duration of that inhibition, where 600 milligrams, this gives you a longer effect. So as we think about the pharmacokinetic exposure and PD effects that we observe, I think it's important then to step back and say, okay, well, what does this mean in terms of how predictive is this of potential clinical efficacy, and how should we think about dosing moving forward? So I think a useful way of doing that is comparing what we see to approved acute C1 inhibitor treatments that are also working by inhibiting plasma kallikrein. What we can see is with C1 inhibitor potency, it is a relatively weak inhibitor, which is not surprising given that it is the endogenous inhibitor. So the IC50 in whole plasma of the C1 inhibitor is approximately 1,700 nanomolar, so it's pretty weak. KVD900 is 20 times more potent at 90 nanomolar. If we used the approved drug Berinert, and we basically from its package insert take its approved dose, at 20 units per kilogram and look at its maximal concentrations of 3,000 nanomolar that's reached within 48 minutes of dosing, we can come up with a maximal concentration of Berinert at 2 times IC50. And you can do this with the other C1 inhibitor preparations, and you come to a very similar answer. So this 2 times IC50 number is important now as we think about KVD900 as we move this forward into clinical trials. So again, using that bar, this 2 times IC50 bar here, we can look at the pharmacokinetic curves of both 600 here and 100 milligram here of KVD900. And what you can see is very rapidly, if you can call it kind of the magic threshold here, we cross that 2 times IC50, and we quickly get above 10 times IC50 and even 100 times IC50. With the 600 milligram dose, we get to 100 times IC50 in less than 30 minutes. So this is really important, I think, as we think about the potential for efficacy of the drug. Also really important in this particular indication for acute therapy is how quickly we get there. One of the things we were often asked before we had generated data with 900, and we were talking about this conceptually, was, well, how quickly can you really get an oral drug on board, and can that compete with injectable therapies? And I think what this panel, this bottom panel, tells you is yes, we can. This is actually the same data as you see from above, just with the x-axis just focused in on 120 minutes. And what you can see is we cross that 2 times IC50 in about 10 minutes after dosing, which is, I think, pretty impressive, and then get up to 100 times IC50 in about 30 minutes. So for us, this is really important as we think about our potential for clinical efficacy and clinical utility. So the data that I've just shown you, we believe, represents a potentially ideal profile for an acute oral therapy for HAE. The tablet, the tablet data is even more impressive than the capsule data we generated before, with even faster and higher absorption and effective concentrations for out to 10 hours after dosing. We saw no significant GI adverse events at all, and that would certainly encourage this moving forward, as that's, I think, an important characteristic of the drug. And we minimize our tolerability findings by dosing acutely and having the drug have a short residence time. We think that 900 has the potential to be a really patient-favored drug, or the patient-favored drug for the acute opportunity. And one of those reasons is because injectable therapies, while very effective, are often compromised by the delay in treating these attacks. And that delay is due to a number of factors, certainly convenience and availability, as well as pain, factor into when patients take their drug. And so I think as we look at some of that data, and this is from the Academy Outcome Survey, this is Firazyr, and this is actually a pretty robust study in over 207 attacks, where we're looking at attack duration by time of initiation of treatment. And what we can see here is that if we intervene early with Icatibant in the first hour, we have a relatively short duration of the attack, so we can shrink down that attack duration to approximately 5 hours. However, if we delay treatment out here to greater than 5 hours, effectively you have a 24-hour period of attack. So what this says is intervene early. That's really critical. So that's great. So if everybody with an acute treatment, with an injectable acute treatment intervenes early, this shouldn't be much of a problem. But what we can see is that doesn't happen. And this is a controlled clinical study, we see that just approximately 40% of patients are dosing within that first hour, and actually 30% of patients are dosing greater than 5 hours later. And again, as I mentioned earlier, why is this? It's certainly more inconvenient. It's a challenge to find your to locate your preparation, to administer it in a private place. It's very painful, and I think those factors all combined often lead to a delay, and times lead to no treatment at all. So this is a bit of a cartoon of how we think KVD900 may compete in this market. As I mentioned, we get on board very quickly into even higher levels than what we see with injectables. So we think that has an opportunity to lead to much earlier treatment, which we know improves efficacy, even into the very early or prodromal stages of an attack. And we think this has the opportunity to dramatically improve patient quality of life by providing better disease control and better efficacy when we think about HAE. So we think about commercially, where are we headed? This is the acute market, as I showed you earlier. Certainly, we think we're delivering a profile that we think will be very compelling for these patients who are currently taking injectable therapies. We also see opportunity in patients who are currently on prophylaxis, who with an oral therapy may decide to switch over and with something that is very convenient to take and highly effective. We're currently running a phase 2 clinical trial with KVD900. This is in 50 patients with HAE, approximately 50 patients with HAE. The study will be conducted in Europe, 10 to 15 sites in the UK, Germany, and other European countries. What we're looking for are patients that have a reliable attack history. So patients have to have had at least 3 attacks in the previous 90 days. And the study will be broken into 2 parts. First part of the study will actually be in the clinic when patients are not experiencing an attack, so in an intercritical period, where we're giving them an open label dose of KVD900 and looking at pharmacokinetics and pharmacodynamics in patients. From there, everyone will progress on, so there's no selection based on part 1. Everyone progresses to part 2 of the study. And this is a randomized sequence of both KVD900, 600 milligrams, and placebo. So what happens is patient takes home 1 treatment. They experience their first attack. They call their physician. They confirm an attack has started, and they dose. We then monitor symptom severity and use of standard of care medication following that dosing. They then come back to the clinic and are crossed over to receive the alternative treatment and then do the same thing with their second attack. Importantly, we're looking to dose early, and we talked about that, the importance of dosing early. So we want them dosing within 1 hour of attack, and then we'll monitor symptom severity for 24 hours following dosing. The primary endpoint will be an evaluation of the use of standard of care between the groups, as well as we'll evaluate symptom severity scores using visual analog scale, as well as other categorical so-called Likert scales as we evaluate various elements of the severity of the attack. This study will read out in late 2019. Switching gears now to talk about our work in diabetic macular edema. In contrast to HAE, DME is unfortunately a very, very prevalent disease. There's approximately 900,000 patients in the world that are at serious risk of vision loss. The disease is characterized by retinal swelling due to leaky blood vessels in the macula. It's a leading cause of blindness in the developed world. The standard of care is intravitreal administration of various anti-VEGF agents. There are currently no oral treatments available for DME. But importantly, a significant proportion of patients, and I'll come back to this, do not respond well to these anti-VEGF therapy. And that really distinguishes DME from other diseases where we use VEGF inhibitors such as AMD, where you have a much higher response rate to those therapies. Plasma kallikrein has been identified as a potential VEGF-independent mediator of diabetic macular edema. This has now been well published over the past several years, and I think we have a good understanding of the mechanism preclinically and why it should be important in DME. KalVista has developed KVD001 as an intravitreal therapy. We'll talk about that drug. It's now in a phase 2 clinical trial. We're also looking to bring forward oral therapies for DME. We signed a collaboration with Merck in October 2017, and our work in DME is covered in that collaboration with Merck, and I'll talk about that in just a few minutes. I referenced earlier the response rates in DME, and this is some data from the DRCR Protocol I study. This is one of the largest studies we have actually over multiple years of use of an anti-VEGF. This is actually Lucentis in this case. And what I'll draw your attention to is the differential in the data we see from these patients. The top line of data here, again, the y-axis is change in letters, so this is a vision indicator, as well as time on the x-axis. What we can see with this top line of data, I would submit to you, is what we think of the classical response to an anti-VEGF. So kind of a 15-letter or 3-line gain in responder population, just over a third of the patients. In the other groups, though, what we can see is a more middling response here, with approximately 6 letters and about a quarter of the patient. But importantly, what we're focused on is this bottom line of data of approximately 40% of patients who don't respond at all. Each one of these small dots as we move across here is an intravitreal injection of anti-VEGF. So we're really not seeing a response to therapy. And these patients, we think, or hypothesize, may be, their disease may be mediated through a different mechanism, perhaps plasma kallikrein. This is just one piece of the preclinical data package that I mentioned earlier. And what we're looking at here are samples, vitreous samples from patients who have DME. These samples are, as you might imagine, from the back of the eye. This is really difficult to get these types of samples. We obtained these actually from a cohort of patients in Japan who had vitrectomy, so we had access to the vitreous samples. But what we've done in each patient is we've looked at levels of both plasma kallikrein and VEGF. So again, each number along the bottom represents a patient. And what we've done is we've ranked these samples in increasing levels of VEGF. So the black bars are levels of VEGF in each one of these patients. What you can see very quickly is the levels don't correlate between plasma kallikrein and VEGF. And I think from our perspective, what's really interesting is you see a group of patients here on the left-hand side who have very low levels of VEGF, but correspondingly high levels of plasma kallikrein, perhaps indicating that these patients may not be the ones you would expect to respond from a VEGF inhibitor, but may be responsive to an inhibitor of plasma kallikrein. Here's data from our phase 1 trial from KVD-001. As I mentioned, 001 is a small molecule plasma kallikrein, an intravitreal approach here. This is a first-in-class therapy. This is the first clinical data we have in humans with a plasma kallikrein inhibitor injected directly into the back of the eye. This study, because of that type of administration, was conducted in patients. These are 14 patients over 3 doses, three patients at the low, 3 patients at the mid, and 8 patients at the top dose. All the patients had previously received treatment with anti-VEGF therapy, but had come off that therapy to enter into the study. So presumably were not having a response to therapy because otherwise they wouldn't come off. The drug, importantly, from the study was well tolerated, and from our perspective, as a first time in humans injecting drug into the back of the eye, we were thrilled with that. What you can see here is data, and again, I'll just caution, this is not a controlled study, right? So we don't have a control group. But what we've done is we've just looked at the mean change in vision across the study, meaned across 3 doses. I think pretty impressively, you see at day 84, a mean improvement in vision of about 4 letters across the 3 doses. This isn't a proof of efficacy, but it certainly is a signal that we thought was consistent with the preclinical data we had and was intriguing as we thought about moving forward. The preclinical data and the phase 1 data were certainly part of the package that we discussed with Merck when we signed our collaboration with them in late 2017. The collaboration is structured as 2 distinct options, one over the intravitreal program and one over our oral program for DME. We run the programs until the time Merck exercises its options. And we are responsible for clinical development and preclinical development of the assets. Once Merck exercises, they pick up all the costs moving forward and control the program. Merck paid us $37 million up front for the program, for the option agreement, as well as they also did a $10 million pipe with us and acquired just under 10% of the company at the time. There's an additional $715 million of payments to come. Those haven't been disclosed in terms of the milestone tiering, but I will say that there are significant payments coming on the exercise of options as we move forward and milestone payments moving forward. In addition to those payments, we have tiered sales royalties on global net sales, and as I mentioned earlier, the pipe that Merck acquired at the time we signed the deal. Just briefly now on the phase 2 clinical study for 001. This is a very large study in DME with approximately 123 patients who are that group of patients that I referred to earlier who have discontinued treatment with anti-VEGF These are patients, we require that they had a good course of therapy with anti-VEGF before coming into the trial, and these patients still have significant edema and reduced visual acuity despite those treatments, having been through that trial of treatment with anti-VEGF. So again, trying to focus in on this patient population that we think may benefit the most from plasma kallikrein and perhaps even enrich for response to plasma kallikrein. It is a sham-controlled, double-masked clinical trial that will evaluate 2 doses of KVD-001 compared to a sham control. The primary endpoint will be a look at vision, best-corrected visual acuity, as well as other endpoints including central subfield thickness, or CST, and other diabetic retinopathy outcomes in the trial. What we're doing is now we've moved from a single dose, as we did in phase 1 now, to multiple injections. We're having 4 injections over 3 months of treatment at day 0, 30, 60, and 90. And then we'll have a 3-month follow-up following the dosing period. The study will complete in the second half of 2019. We're pleased with the status of the enrollment and our ability to complete the study in that time frame. Just finally now, just to summarize the milestones coming up for the rest of this year. With A24, we will give an update on this program mid-year. This is our first in-human trial, and give some indication of which indication we'll be pursuing with A24. We'll complete our phase 2 trial with KVD-001 in the second half of this year, and in late 2019, we'll have data. We expect to have data for KVD-900 from its phase 2 clinical trial. And with that, I thank you for your time. Any questions I can answer?
I think there's a couple other oral HAE trials that are currently in pipeline.
Right.
What would you say would be the biggest differentiator for KVD-900?
Sure. I think that the one I would highlight is the fact that KVD-900 was prospectively designed to be an oral acute therapy for HAE, right? So we kind of sought to have the profile that we've actually achieved, which is very rapid and high exposure of the drug, because we think that is ultimately what will lead to the most success both in a clinical trial setting as well as commercially. So I think when you compare the profiles of the drug, how quickly the drug gets on board to what exposures you see, I think you'll start to see some pretty important differences there. And I think it goes back to kind of first principles of kind of what we sought out to achieve and the indication we're focused on with KVD-900. Okay, thank you very much.